13.3. If you're wondering why I'm dressed like this, this is Halloween. People who are watching this.
Who are you?
I'm Carl Spackler from Caddyshack. He's like the maintenance guy. It's my favorite movie. Okay, page 247.
How'd you fix it?
Couple videos I didn't get to show you yesterday because I didn't have them on here. So we'll look at them. This is about bar bodies. Do you remember what a bar body is?
Inactive
Chromosome.
An inactive X chromosome. So there are the two X chromosomes. Let's see what they say.
XInactivation is a process that mammals use to equalize the gene dosage between males and females. Since normal male mammals have only one X chromosome and females have two, there is a potential imbalance in the expression of genes on the X chromosome between males and females. In female mammals, this imbalance is prevented by inactivating all but one X chromosome in all somatic cells. XInactivation can be divided into three phases: initiation, spreading, and maintenance. Initiation occurs during embryonic development. Each X chromosome has an XInactivation center, or XIC. The number of XICs are counted, and one X chromosome is randomly chosen to remain active while the other is targeted for inactivation. Spreading is the process of compacting and inactivating the X chromosome. A gene designated XIST within the XInactivation center encodes an RNA that coats the X chromosome and promotes compaction, beginning at the XIC and progressing toward both ends until the entire X chromosome is inactivated. When spreading is finished, the compacted chromosome is called a bar body. After the bar body is formed, maintenance begins and continues through adult life. The bar body is replicated just like other chromosomes, but both copies of the replicated bar body remain highly compacted and essentially inactive. Thus, once an embryonic cell undergoes XInactivation, all cells that derive from it will have the same X chromosome inactivated.
Huh.
So it's just random?
It's random which ones get picked in the embryo. Yeah. But once they copy themselves, it's the same as the one that...
What was the last extra credit? Was it coffee?
I don't remember.
Is the bar bodies the same thing in like dogs and like all animals?
All mammals do this. All female mammals.
Tortoiseshell cats have different
So,
colors.
What about like dogs that have like a brown eye and like?
That's probably not this. It just so happens that...
Not like a brown eye, but like the fur around it.
That's probably not this. It just so happens that on cats, there is a gene for coat color on the X chromosome. You got to remember there's you know, a bunch of other chromosomes that carry information too, other than just the X. So the gene for fur color in dogs is probably just not on the X chromosome, so you don't see tortoiseshell type dogs. It's only on the X chromosome in cats, in the particular type of cat.
So not all female cats just that one?
For tortoiseshell?
Yeah, but I mean, could it rand? Why are cats solid colors?
Well, not all cats are that. I don't know. It's not species, but it's
Breed?
that breed of cat. Only that breed of cat has those genes on the X chromosome. See what I'm saying? And even tortoiseshell cats, if you're homozygous, then if you have two orange genes, then you're going to be orange. If you have two black genes, then you're going to be black. It's only when you have an orange and a black that you end up tortoiseshell. We like cats?
I love kitties.
How many cat people in here?
I have two. I have a cat.
How many dog people in here?
Oh, did I write them? Yeah.
I like cats.
About even?
Which ones? I'm scared. I'm scared of cats.
You scared of them?
They're everywhere in our neighborhood.
Like I'm really
Everywhere on this island.
Okay, let's talk about.
Everywhere.
Let me show you. This next video is about transcription factors. Transcription factors, listen, they're proteins that have to stick to the DNA before transcription can occur. So let's watch these proteins happen.
For RNA polymerase to successfully bind to a eukaryotic promoter and initiate transcription, a set of proteins called transcription factors must first assemble on the promoter. The assembly process begins upstream from the transcription start site, where proteins called basal factors bind to a short TA-TA sequence in the promoter. Other basal factor proteins then bind, eventually forming a full transcription factor complex able to capture the RNA polymerase. Basal factors are essential for transcription, but cannot by themselves increase or decrease its rate. A second set of transcription factors, called coactivators, link the basal factors with a third set of transcription factor proteins called activators. Activators are regulatory proteins that bind to sequences on DNA called enhancers. Enhancers are located at sites that are distant from the promoter. The interaction of activator proteins with transcription factor subunits increases the rate of transcription. Many enhancers scattered around the chromosome can bind different activators, which provide a variety of responses to various signals. When a second kind of regulatory protein called a repressor binds to a silencer sequence located adjacent to or overlapping an enhancer sequence, the corresponding activator is no longer able to bind to the DNA.
So all of this is going on to allow transcription to occur. And remember, transcription occurs, y'all understand this is RNA polymerase. This is the thing that's going to bind to the promoter and do transcription. So in my little model here, the RNA polymerase binds to the promoter and does transcription. Well, in a eukaryotic chromosome, there's a whole lot of other things that got to go on before that RNA polymerase can bind and do transcription. All these factors have to be in place. And if they're not, you don't get transcription of the gene. So there's a wide level of control that goes on. Do you see? That section 13.2 is really important. Maybe because it's Halloween coming up today, maybe you didn't get to do the reading. Make sure you read that section. They do ask a lot of questions about control of making proteins. That's big in the minds of the test makers. Okay, so what we're going to do now is we're going to talk about mutations, which is really starts on page 247. I have this out of order.
What page?
No, it's after this. Okay. It starts on 247, page 247, 248. Basically, a mutation is when the DNA, something happens to it, where the right base isn't in the proper place. For instance, when you're copying the DNA, you remember when we were copying the DNA, we had helicase came in here and split it up, and then you're putting on new nucleotides. And here we're coming in with new nucleotides and putting them in place. You remember this process? DNA polymerase was an enzyme that did it. Are y'all with me here? Well, sometimes the wrong nucleotide can be put in place, even though they don't match up perfectly. And what happens is, see this, the DNA polymerase enzyme, which was this one. We'll go ahead and throw DNA helicase too. DNA helicase unwinds it. And DNA polymerase, just one by one, puts them on. Click. 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 And then, whoops, that one doesn't fit. That's the wrong one. It put a G instead of a T. Well, that can end up causing problems. Now, normally what happens after the DNA polymerase is finished, there is a proofreading enzyme that goes around and checks to make sure everyone is correct. And it just goes along and checks. But even the proofreading enzyme can mess up every now and then. And it just so happens that about 1 in a billion nucleotides gets put in wrong every time a cell divides. And you think, well, that's pretty good, you know, it's like a copier running a billion times and only messing up one sheet. You know, that's pretty good. That'd be pretty good for a photocopy machine. But what happens is these mutations, so now you got the wrong letter here, and it's kind of like if I wrote a sentence, and this is the way it's often taught, kind of a good way of teaching it. Let's write a sentence using only 3-letter words. Now remember, the DNA code is read 3 letters at a time. So I want you to read this sentence 3 letters at a time.
The cat ate the rat.
The cat ate the rat. Makes perfect sense, doesn't it?
Yeah.
Well, if we screw up 1 letter, then you have the eat ate the rat, and it doesn't make sense. What is the eat ate the rat? I don't know what that means.
Mutation.
So that's a mutation. And what that would end up doing, remember, here's our code that was built 3 from 3 letters at a time. These 3 letters coded for that amino acid, these 3 coded for that amino acid, and so forth. What a single mutation like that would do, we call this a point mutation. Point mutation is it would throw off the code here. So instead of a methionine right here, we might have a tryptophan.
A point what?
A point mutation. So in our long protein chain, where's my protein? Don't you hate it when you lose your protein? Here it is. In this long protein chain, if we had a point mutation like that, one of the amino acids is screwed up. So what will that do to our protein?
Change it.
How would it change it?
To make it completely different protein.
Well, it may change it. It may change it. And so remember, protein folds a certain way, folds into a certain shape. And the way it folds depends on the amino acids. Different amino acids have different R groups. And sometimes the R groups are charged. They're like magnets. So if let's say the green ones are charged R groups, act like magnets, then this green one might stick to an orange one, let's say the orange is positively charged and the green is negatively charged, then these charged R groups will stick together, the green and the orange will stick together. And that affects the way the protein folds up, these charged R groups. If the mutation occurred where it changed, it took away a charged R group, you wouldn't get that binding anymore when the protein was folded up. And it would fold into a completely different shape, and now your protein wouldn't do the same function that it used to do. Because enzymes and all proteins work based on their shape, you see. So by replacing a single base, we could replace a single amino acid, and that could change our protein. Now, often it doesn't. Often a mutation will change an amino acid and not change the function at all. Like you could put a pink here, or you could put a white or a blue, and you'd still get the same shape protein. So sometimes it doesn't affect the protein, and sometimes it does. Sometimes, let me go back here a little bit. Man, this is a long one. Oh, wait, I need to go to a different chapter. Enable. I'm an enabler.
It's not always a good thing.
You know, when I was a kid, we didn't even have computers. You had to learn on a chalkboard. It's a wonder we learned anything. Okay. Go to this chart here. Remember this chart?
Yeah.
This chart tells what amino acid a sequence gives. Like GUU will stand for valine. But if we screw up and we get a mutation that changes the GUU code to GUC, guess what? We're still going to get valine as the amino acid. You can have a mutation that doesn't even change the amino acid. We call these silent mutations. And so there's a good chance that even when there is a mutation that it's silent, or it doesn't even affect the protein. And there will be no change in the organism. This is important when we study evolution. You know, the Stovall family could have come up with a mutation in their genes over the years. Maybe your great-great-granddad had some mutation in his sperm cell that changed one of the letters, and that will make your DNA different from my DNA. But it wouldn't affect our body chemistry at all if it is a silent mutation that just changes one of these letters that doesn't even mean anything. And so you can actually chart, you can look at the DNA and see how different Stovall DNA is from Willis DNA. And you can use these numbers of mutations, even though they're silent, will give you a measurement of how long ago our two families split in the evolutionary line. You see what I'm saying? I mean, all of us are related. Did you know that? If you go back far enough. If you go back far enough, every human being is related. Every organism is related. Actually, at one point on Earth, there were just a few bacteria, and every organism came from those. We'll talk about that in later chapters. So you could these regular mutations give you kind of a measurement of evolution, which is kind of a cool thing. Oh, let me give you another. What if this happened? What if we were going along? What if? Where's my DNA polymerase? Here it is. What if DNA polymerase is going along and it doesn't put in a base at all, what if it leaves it out?
And it would
Well, then you
change
can have
all
a-
of the groups. that code
This is
on.
a catastrophic mutation. Love that word, right?
Does it change the base at all?
Yeah, what if it just leaves one out?
Then it would change the.
Then let's just leave out a letter. And I want you to read this 3 letters at a time, because remember, the cell doesn't think, so it just reads 3 letters at a time. The atta tet her at. The atta tet her at is a completely, it sounds like a foreign language there. We call this a frameshift mutation. A frameshift mutation. The same thing would happen if we inserted a letter somewhere. So let's go back to the cat ate the rat. If we insert another C right there, then it's the kuka tot eth era. So if you insert an extra base, or you take out an extra base, that's called a frameshift mutation, and it doesn't just change one amino acid, it changes every amino acid after that. And so my protein would be completely different just by one little change. And like I say, it only happens 1 in a billion nucleotides. But since there's 6 billion nucleotides in the entire human genome, that's 6 screw-ups every time a cell divides. Skin cells might divide 10,000 times in your life. That's 60,000 screw-ups. Now you've got problems, and your skin looks weird. You ever look at an old person's skin compared to a young person's skin? Doesn't look so good. It's like taking a photocopy and copying it over and over. Pretty soon it's going to start looking bad. You know what? You know some of the things that cause mutation?
Carcinogens.
Carcinogens. You can have chemicals. You got to avoid carcinogenic chemicals, because there are chemicals that get into the nucleus and screw with the way this thing works. We call them carcinogens. Another bad thing that causes mutation is radiation, the sun. You lay out in the sun, you'll get a bunch of mutations, because solar energy goes in here and strikes these molecules, and while this is moving along nicely, if it's hit by some solar radiation, something might happen. Or the solar radiation might knock one of these pieces out, or something like that. You ever look at the top of your arm and compare it to the bottom of your arm? Y'all are probably too young. All your skin looks pretty good. But if you take an old person like me, the top of the arm, the skin looks a lot worse than on the bottom. That's due to sun. If you protect your skin, you'll have real good skin at an older age. So protect your skin. You won't get cancer as easily. Oh, well. Okay. Lee, you have to go to the nurse.
Lee, you're sick.
You're sick. You have a mutation.
What is it?
They didn't say. They just
Why
said you
do
have to
I-
go.
Do I have to go to the nurse?
No?
What can you say with the point mutation again?
A point mutation is when you change one of the letters of the DNA code. You change one of the bases from one thing to another. That could affect an organism or maybe not.
So just a normal gene mutation?
Yeah. Have you ever noticed a black person has black people have nicer skin when they get older?
Yeah.
You ever notice that? That's because they have sun protection in their skin. They have melanin, which protects you from the sun. If you're real light-skinned and you're in the sun a lot, you're going to have a lot more skin damage.
I won't get the rickets though, that's for sure.
Well, that's true. The vitamin C.
What a rickets.
Rickets is when you don't get enough vitamin D.
Or D, yeah, vitamin
And
D.
your bones mess up. That's why Europeans have light skin, because when they got to Europe, they were dying of this.
This is, the book shows a little test. It's called the Ames test. The Ames test is to test to see if a chemical might be a carcinogen. And so what they do is they take this bacteria and they put it in a test tube. And so what they have growing in this test tube is a bacteria, little tiny organism. And this bacteria has to have histidine to be able to grow. Histidine is one of these amino acids. And so this bacteria won't be able to grow well unless you give it histidine. And these two plates don't have histidine. So the bacteria shouldn't grow on the plate. And that's what happens if we pour our control bacteria onto this plate, we see no growth. But here we take a suspected chemical, some chemical, let's say they come up with some chemical that's good at killing ants. And they say, before we start selling this to everyone in the world, we need to see if it's a carcinogen, if it causes cancer, if it causes mutations. Because mutations lead to cancer. And they put a few drops of it into the test tube, and lo and behold, you pour that on a plate, those bacteria are able to grow, even though the control bacteria didn't. That means that chemical caused some kind of mutation that allowed them to grow, where they normally wouldn't be able to. So they mutated where they could grow without histidine, you see. And so that's one of the tests that they do to see if a chemical is a carcinogen. They don't give it to people and see if they get cancer. That wouldn't be ethical. But so they do this instead. And so if it causes mutations in organisms, it's very likely that it can cause cancer, and then it'll be labeled carcinogenic. And then if it's labeled carcinogenic, you may not be able to sell it to everybody. There are a lot of things out there, though, that are carcinogenic. There's, cigarettes, smoke is very carcinogenic. A lot of the chemicals they use to put in fertilizers and things like that for plant growth and such could be carcinogenic. There's stuff in the foods that you eat that actually preserve the food that you eat that is a little bit carcinogenic, and you shouldn't have too much of it. They found some of the artificial sweeteners that they use can be carcinogenic at high levels.
In lab rats.
Right. Again, they aren't going to do the experiments on humans.
Yeah. Well, it's just funny.
Sometimes it's only in huge amounts is it carcinogenic at all. And you don't you don't eat that much sweet. So, but still, it's kind of something to think about. And if you don't have preservatives for food, then the food goes bad, and you get botulism when you eat the food from the food being spoiled. And that's no good either. So we have to balance these things. We have to come up with some preservatives for foods, or everyone will starve. We won't have enough food for everyone. So you can't just say, get rid of all the preservatives. Then you're killing lots of people from starvation. Have you ever heard of sickle cell anemia?
We talked about this.
Yeah, sickle cell, this is a normal blood cell on the left, and a blood cell that's sickled or shaped funny on the right. And that comes from a single mutation. And they show it here. There's a mutation right there in this base. After the GT, that base, if it gets changed from a G to an A, well, you don't get any change, because that still codes for the same amino acid. It still codes for histidine.
mutation of one nucleotide?
A mutation of one nucleotide, yeah, a mutation of one base, one nitrogen base there. So if it changes from a G to an A, you don't get any difference. But if it... Oh, here we go. If we get this change over here, if you change this base, you get a change in the amino acid from glutamine to valine. See, the amino acid has changed. And that will screw up the protein. And this protein happens to be hemoglobin. Hemoglobin is a protein that's in your blood cells that carries oxygen. And if you get this mutation, the blood doesn't carry oxygen very well. And you can end up with sickle cell anemia. Here's a really bad screw up. If it changes so it codes for a stop codon. You remember what the stop codons were?
Yeah.
UGA was a stop codon. What if you get a mutation where it changes from CGA, which codes for some amino acid, to UGA? Then you'll get a stop, and the protein will be much shorter. We call that a nonsense mutation. A nonsense mutation is when the mutation codes for a stop.
Makes no sense.
Yeah, and you get a protein that makes no sense. It's too short. We did talk about, though, that people who are heterozygous for sickle cell anemia are malaria resistant, right?
Yeah.
Remember that? So if you're heterozygous, you have two genes, one for sickle cell and one for normal blood. Well, then you actually don't get sickle cell. You make normal blood for the most part, but you can't get malaria. It takes having two sickle cell genes on the two homologous chromosomes to actually have sickle blood cells.
Is sickle cell recessive?
It is.
Weird.
Yeah. That's why you have to have two of them. If it's dominant, you only need one of them to have the disease.
What's that called? The stop?
This is a...
The pink thing that stops it in a nonsense mutation.
Stop codon.
Stop codon.
That's the
on the board, the pink magnet on the board.
Oh, this thing, that's a releasing factor is what we call it.
Okay.
That binds and ends the whole thing. Releasing factor. Let's watch this video
A
on
mutation occurs by base substitution when an incorrect base is incorporated into DNA. Some base substitutions occur because purines and pyrimidines exist in two structural forms. The most common form results in base pairing between adenine and thymine and between guanine and cytosine. However, the hydrogen atoms can move to form a base with altered hydrogen bonding properties, creating a tautomeric shift. When a tautomeric shift occurs in adenine, the adenine can bond to cytosine. A tautomeric shift in thymine allows it to bond to guanine. This error in DNA replication is passed on to the cell's progeny. The change in a single nucleotide in the DNA results in a change in the corresponding nucleotide in messenger RNA. The change in the codon can result in a different amino acid being incorporated into the protein.
So when the cell was copying itself, it screwed up and put the wrong amino acid in place. This pair is supposed to be AT, put the wrong base in place. And it got changed to GC after a couple divisions. Now you can see that if we put the wrong one in place right here, I'll leave it sticking out a little bit. That's a screwed up pair. But if it divides again, then and is placed normally, then this was the wrong base, but it will now pair with the correct. This was a guanine. It'll have a cytosine next to it and be a normal strand after the second division. So you don't see anything abnormal after two divisions.
nucleotide sequence in DNA determines the nucleotide sequence in messenger.
Let me just run this through here. There's a mutation. Look, they've added an extra C in place.
What kind of mutation is that?
What do y'all think? What's it called when we add an extra?
Frameshift.
That's a frameshift mutation.
Well, it left
And it
the same-
screws everything up after it.
What's the other? What do you call it? Frameshift and what?
A point mutation?
Like,
A point mutation only screws up the one base it codes for.
that's the catastrophic one.
Yes, it changes everything.
So if extra codon was added to or deleted from.
Yeah, because I want the frameshift one to completely different. Because it leaves it out or adds it on.
Yeah, check this out.